Dive into the mystery of type O blood, an ancient whisper in the veins of the Americas' indigenous peoples. This enigmatic marker, more than just a type, unveils a story of survival, adaptation, and deep-rooted connections to the land. What secrets does it hold? Join us as we delve into the mystery of type O blood. Chapter 1. Blood Basics. Blood, our vital life force, flows intricately and complexly through our veins. Its importance stretches beyond simply keeping us alive. It weaves a rich narrative of history, genetics, and evolution. Central to this intricate story is the ABO blood group system, dividing human blood into 4 primary types: A, B, AB, and O. Each type hinges on the presence or absence of specific antigens on our red blood cell surfaces. These antigens, which our immune system can identify and possibly react to, are proteins and sugars that decorate our red blood cells. Types A and B possess their respective antigens. AB houses both, and O, the mystery in our tale, carries none. O blood, often hailed as the universal donor, lacks A and B antigens, minimizing the chance of rejection in recipients with differing blood types. This trait renders O blood exceedingly precious in emergencies and transfusions when the recipient's blood type is unknown. But why is O blood predominantly found in the indigenous peoples of the Americas? Part of the answer lies in the evolutionary benefits it might have provided. Without A or B antigens, O blood carries a reduced risk of hosting certain diseases, potentially offering a survival edge. Furthermore, some studies suggest O blood may offer more protection against severe diseases like malaria, although this theory aligns more with blood type distribution in Africa than the Americas. Exploring the distribution of blood types among different populations reveals a captivating tale of human migration and adaptation. Globally, type O is the most common, yet its prevalence significantly differs by region. In the Americas, especially among indigenous groups, the proportion of type O blood is remarkably high, indicating a strong evolutionary selection for this characteristic over thousands of years. The exact reasons for this preference remain somewhat elusive but likely involve a mix of environmental challenges and disease resistances where type O blood had an edge. One theory suggests that type O blood's supposedly superior oxygen delivery at higher altitudes may have given ancient inhabitants of the Americas' mountainous terrains a distinct survival advantage. This intricate tapestry of blood types paints a fascinating picture of our past, underscoring the deep connections between our biology, environment, and history. Chapter 2. Geographic Bloodline Mysteries. The global distribution of blood types presents a fascinating mosaic of human migration, adaptation, and the mysteries of ancient populations. Among these patterns, the distribution of type O blood, especially its pronounced prevalence in the Americas, stands out as a particularly intriguing enigma. This chapter delves into the geographic spread of blood types, comparing the unique concentration of type O in the Americas with its presence around the world, unraveling the complex tapestry of human history and movement. Blood type distributions vary significantly across the globe, reflecting millennia of human evolution, migration, and interaction. While type O is the most common blood type worldwide, its prevalence is not uniform. In some regions, it dominates, while in others, A, B, or AB types are more common. This variation is key to unlocking the secrets of ancient human migrations and interactions. In Africa, the cradle of humanity, type O is highly prevalent, supporting theories that it is the oldest human blood type. Moving into Europe and Asia, the prevalence of type O decreases, with type A becoming more prominent in Western Europe and type B in Central and Eastern Asia. This shift hints at the complex interplay between genetics, environment, and social structures that have shaped human populations over tens of thousands of years. The stark contrast in the prevalence of type O blood in the Americas, particularly among indigenous populations compared to other regions, poses compelling questions about ancient migrations and population dynamics. In many indigenous communities, type O reaches frequencies as high as 90 to 100%, a stark contrast to the global average of approximately 62%. This unique distribution pattern is a crucial piece of the puzzle in understanding the peopling of the Americas. Theories on how type O became so prevalent in the Americas often center on the Bering Land Bridge, which ancient populations are believed to have crossed during the last ice age. These early migrants, carrying predominantly type O blood, would have been the ancestors of today's indigenous peoples. The subsequent isolation of the Americas from Eurasia until the arrival of Europeans could explain the maintenance and increase of type O prevalence due to genetic drift, founder effects, and selective pressures unique to the environment and challenges faced by these populations. The isolation of the Americas for thousands of years before European contact is a key factor in understanding the geographic distribution of blood types. This isolation would have limited the introduction of new genetic variants, including blood types, allowing type O to become more entrenched in the gene pool of indigenous populations. Additionally, the challenges and selective pressures faced by these populations, such as diseases and environmental factors discussed in previous chapters, may have further favored individuals with type O blood. Comparing the Americas' type O prevalence with that of other isolated populations worldwide offers additional insights into the impact of migration and isolation on blood type distribution. For example, some Pacific islands and remote communities also show high prevalences of certain blood types, reflecting their own unique migration histories and periods of isolation. However, the scale and consistency of type O's dominance in the Americas are unmatched, highlighting the unique historical and prehistorical context of these continents. This distinction raises fascinating questions about the movements of ancient peoples, the routes they traveled, and the survival strategies they employed. Chapter 3. Tracing the Type O Blood Trail. The odyssey of Type O blood through the annals of history and across the expansive landscapes of the Americas is a narrative filled with mystery, resilience, and evolution. This chapter ventures deep into the heart of the enigma, piecing together historical and scientific clues to understand why Type O blood flows so prominently in the veins of indigenous peoples across this varied continent. Our story starts with the peopling of the Americas, an epic migration saga spanning thousands of years as early humans trekked across the Bering Land Bridge from Asia into the Americas during the last ice age. They carried with them genetic markers destined to influence the bloodlines of their descendants. Among these was the O gene, a silent beacon of survival and adaptation. Scientific exploration into the distribution of blood types among indigenous populations has uncovered intriguing patterns. For example, a pivotal study in the 1980s with Native American populations in Arizona revealed a remarkable finding. An astonishing 99% of participants had type O blood. This significant occurrence is mirrored in indigenous communities from the snowy peaks of Canada to the verdant rainforests of the Amazon. But why is type O blood so prevalent among these groups? The answer may be found in the crucible of natural selection and environmental adaptation. The early settlers of the Americas endured extreme conditions from the icy tundras of the north to the thin air altitudes of the Andean highlands. In these challenging environments, type O blood could have provided a vital advantage. The lack of A and B antigens on red blood cells may have lowered the risk of infectious diseases, which were widespread and often deadly. Additionally, the adaptability of individuals with type O blood to various environments might have solidified this blood type's dominance as indigenous peoples dispersed across the continent. Those with type O blood were possibly more adept at surviving and flourishing, passing their genetic legacy through generations. This selection process, unfolding over millennia, would naturally result in the significant presence of type O blood seen today in indigenous populations. However, the story of type O blood is not just one of survival against the elements and diseases, it is also a tale of human connection and shared heritage. The prevalent type O blood among indigenous groups acts as a biological link, uniting disparate communities across the continent. This commonality hints at a deep, shared ancestry that transcends the modern boundaries that divide the Americas. Recent advancements in DNA technology have shed light on the journey of type O blood through time, allowing geneticists to trace lineages and migrations with remarkable accuracy. These studies confirm the ancient roots of type O blood in the Americas and its significance as a marker of indigenous heritage. Yet, the historical journey of type O blood is not merely a subject of academic interest. It has profound implications for health and medicine among indigenous populations. The universal donor status of type O blood means it is vitally important for blood transfusions and emergency medical care. This highlights the critical need for blood donation within these communities where type O blood is highly sought after but not always readily available. Chapter 4. Modern Implications of Ancient Blood. Tracing the journey from ancient migrations to our current era, we uncover that type O blood, rooted deeply in history, holds significant implications for contemporary medicine and healthcare. As we delve into the mysterious origins and evolutionary benefits of type O blood, its impact on today's medical treatments, emergency response strategies, and the global blood supply becomes increasingly evident. This narrative explores the pivotal role of type O blood in modern medical practices, highlighting its crucial presence in emergency transfusions and the broader implications of its universal donor status. In the realm of transfusion medicine, compatibility between blood types is critical. Type O blood, with its unique characteristics, forms the foundation of emergency medical care and surgical procedures. Notably, type O negative blood is esteemed as the universal donor for red blood cells capable of being transfused into patients of any blood type without the risk of adverse reactions. This feature is indispensable in emergencies where the patient's blood type might be unknown. Moreover, type O positive blood, while not universally compatible, can be administered to any recipient with a positive Rh factor, covering a substantial portion of the population. Such versatility renders type O blood an invaluable asset in saving lives, particularly in trauma care and critical blood loss scenarios. The demand for type O blood, especially O negative, remains high worldwide, from emergency rooms to disaster zones and battlefields. Its status as the first choice for transfusion when the recipient's blood type is uncertain underscores the ongoing necessity for type O blood donors and the importance of maintaining sufficient supplies in blood banks and hospitals. Yet, the universal donor capability of type O blood also introduces challenges, including potential shortages due to its high demand across various medical emergencies and procedures. Balancing the availability of type O blood against the needs of daily medical care and emergency responses poses a continuous challenge for healthcare systems globally. Beyond its role in transfusions, the prevalence of type O blood has broader implications for medical and genetic research. Studies indicate that blood type may affect susceptibility to certain diseases, with type O individuals potentially having a lower risk of coronary heart disease and differing responses to pathogens. These insights prompt research into the genetic and molecular basis of these associations, paving the way for personalized medicine that considers blood type in disease risk assessments and treatment plans. Understanding the relationship between blood type and disease could revolutionize preventive healthcare and therapeutic strategies. The critical role of type O blood in medical practices also brings to light ethical and social considerations, such as ensuring equitable access to type O blood, especially in areas with a lower prevalence of this blood type or in under-resourced healthcare systems. Enhancing blood donation rates, improving blood storage and distribution technologies, and fostering international cooperation are vital steps to address these challenges. Additionally, research into blood type distribution and its implications must navigate sensitive issues related to identity, ancestry, and population genetics with respect for privacy, consent, and the cultural contexts of the populations studied. As we move forward, the ancient origins and contemporary significance of type O blood will continue to shape transfusion medicine, genetic research, and public health policies. Advances in synthetic blood products, blood typing technologies, and genetic engineering might offer novel solutions to the challenges of blood supply and compatibility. Nonetheless, the natural properties of type O blood and its role as a universal donor are likely to remain fundamental to emergency medicine and transfusion practices. Chapter 5. The Evolutionary Advantage. The prevalence of type O blood among indigenous peoples of the Americas, particularly in environments as challenging as high altitudes, is a puzzle that has intrigued scientists for decades. This chapter explores the fascinating theories that suggest type O blood may have conferred evolutionary advantages, enabling ancient populations to not only survive but thrive in some of the world's most demanding habitats. One of the most compelling aspects of the type O blood story is its potential role in high-altitude adaptation. The Andes, stretching along the western edge of South America, have been home to human populations for thousands of years. Living at elevations where oxygen is scarce requires significant physiological adaptations. Interestingly, the prevalence of type O blood is notably high among indigenous populations in these regions, suggesting a possible link to survival in low-oxygen environments. Why would type O blood confer an advantage here? The theory hinges on blood viscosity and oxygen delivery. Type O blood is known to have lower levels of certain clotting factors, potentially resulting in lower blood viscosity compared to types A, B, or AB. This could theoretically facilitate easier blood flow and more efficient oxygen delivery to tissues under conditions of low atmospheric oxygen, a crucial advantage in high-altitude living. Another angle from which type O blood's evolutionary advantage emerges is disease resistance. Historical data suggests that certain diseases may have affected populations with A or B antigens more severely. For example, some studies indicate that individuals with type O blood have a lower risk of severe malaria compared to those with other blood types. Despite malaria not being a primary concern in the pre-Columbian Americas, this characteristic hints at a broader principle. The absence of A and B antigens on red blood cells might offer some protection against pathogens that exploit these molecules to invade or damage host cells. This theory extends beyond malaria. The general idea is that type O blood could have presented a less favorable environment for a variety of pathogens, reducing morbidity and mortality from infectious diseases. In the context of indigenous American populations where isolation could have magnified the impact of disease outbreaks, such resistance would be a significant evolutionary boon. Theories on type O blood's advantages also touch upon nutrition and metabolism. Indigenous diets varied widely across the Americas, from the marine-rich diets of the Pacific Northwest to the maize-based diets of Mesoamerica. Some research suggests that blood type may influence digestive efficiency and metabolism, with type O being more suited to certain diets high in protein and fat, typical of hunter-gatherer societies. While these theories are less directly connected to survival in high-altitude environments, they underscore the complex interplay between genetics, environment, and lifestyle in shaping human adaptation. An often overlooked aspect of type O blood's evolutionary advantage is its potential impact on social structures and reproductive success. In tight-knit indigenous communities where cooperation was essential for survival, the universal donor capability of type O individuals could have had significant social value in times of injury or childbirth complications. Type O individuals could provide life-saving transfusions, indirectly increasing their value within the group and possibly their reproductive success. While each of these theories offers a tantalizing glimpse into the possible advantages of type O blood, it's important to approach them with a degree of caution. The relationship between blood type and evolutionary fitness is complex and influenced by a myriad of factors, including environment, pathogens, diet, and social structure. It's likely that the evolutionary advantage of type O blood among indigenous populations of the Americas arises from a combination of these factors rather than a single definitive cause. Chapter 6. DNA and Ancestry. Unraveling ties in the intricate tapestry of human history, DNA technology has emerged as a transformative tool, unraveling the threads of ancestry and heritage that bind us to our past. As we delve into the connections between blood types and ancient lineages, particularly the prevalence of type O blood among indigenous peoples of the Americas, DNA analysis serves as a beacon, illuminating the paths tread by our ancestors. This chapter explores how DNA technology not only enhances our understanding of human migration and evolution but also rekindles connections to indigenous roots. As exemplified by the story of Sitting Bull's descendant, advancements in DNA technology have revolutionized the field of genealogy, enabling individuals to trace their lineage back through generations with unprecedented accuracy. By analyzing markers in our DNA, scientists can infer ancestral origins, migration patterns, and even historical interactions between different populations. For many, this technology has been instrumental in uncovering indigenous heritage, providing a scientific foundation to oral histories and traditional knowledge that have been passed down through generations. The role of DNA in tracing ancestry is particularly poignant for indigenous communities, where oral histories and cultural traditions often contain intrinsic ties to the land and ancestors. DNA testing offers a complementary narrative, one that can affirm and enrich these traditional connections or reveal previously unknown aspects of an individual's heritage. For descendants of indigenous peoples, this can be a powerful experience, bridging the gap between historical records and personal identity. The ability to identify genetic markers specific to indigenous populations of the Americas has profound implications. It allows for a direct connection to the ancient peoples who first inhabited these lands, offering insights into the rich tapestry of cultures and communities that predate European contact. For many, the discovery of indigenous ancestry through DNA is not just a matter of personal curiosity but a reclaiming of identity and a restoration of bonds that were severed or obscured through centuries of colonization and displacement. This connection is not merely academic. It carries significant cultural, social, and even legal implications for individuals seeking to understand their place within the broader indigenous community. DNA can provide a tangible link to their ancestral past, fostering a deeper engagement with cultural practices, languages, and communal ties. Moreover, for some, establishing indigenous ancestry through DNA analysis can be crucial in accessing resources, rights, and recognition afforded to indigenous peoples. A poignant illustration of DNA technology's power to connect individuals to their indigenous roots is the story of Ernie Lapointe, a direct descendant of the legendary Hunkpapa Lakota leader Sitting Bull. Through DNA analysis, Lapointe was able to conclusively prove his lineage, affirming a familial connection that had been both a matter of personal identity and historical significance. Sitting Bull, a figure of immense importance in American history, symbolizes the resilience and spirit of the indigenous peoples of the Americas. For Lapointe, the DNA evidence was more than just a scientific validation. It was a reaffirmation of his family's oral traditions and their enduring connection to Sitting Bull. This case exemplifies how DNA technology can bridge the past and present, allowing individuals to reclaim their ancestry and heritage in a way that resonates both personally and collectively. While the ability to trace indigenous ancestry through DNA offers exciting possibilities, it also raises ethical considerations and challenges. Issues of privacy, consent, and the potential misuse of genetic information are paramount. Furthermore, the interpretation of DNA results in the context of cultural and tribal identity is complex. Indigenous identity encompasses more than genetic markers. It involves language, culture, community recognition, and a lived experience that cannot be fully captured by DNA alone. Moreover, the commercialization of DNA testing and ancestry tracing has sparked debates about the commodification of genetic heritage, raising questions about who has the right to access, interpret, and profit from this information. Ensuring that DNA technology serves to empower indigenous peoples and respect their sovereignty and perspectives is crucial.